Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us the 189th seminar, and I call it on Institut Pasteur Day. Next Tuesday, we'll have Lunar New Year Day. Happy New Year again. In Asian countries, including China and Korea, Lunar New Year is one of the biggest holidays. For many people, Lunar New Year Day might be a good restart day for New Year's resolutions if their commitments have not been implemented because of procrastination. Even if we fail to do something, it is never late if we start it again. So let's start whatever we decided to do on January 1st. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor David Bikard. He doesn't need any introduction because he's a very well-known scientist and researcher globally, but I will try to introduce him very briefly. He is a professor at Institut Pasteur and also the head of the synthetic biology group at that institute. Importantly, with Professor Jennifer Doudna, of course, a Nobel laureate thanks to CRISPR technology, he started very important, the first microbiome editing Gordon Research Conference 1 month ago, which I had an honor to attend to give a talk and chair a session. That was one of the best conferences I ever attended, and I thank David and Jennifer for their contributions to this scientific community. And also, I thank David for his excellent research and organizing this wonderful conference. David, thanks one more time, and the virtual podium is all yours now, and thank you again.
Well, thank you so much for the kind words, and thank you also for your contributions to the GRC meeting last month. So it's a great pleasure to have the opportunity to take part in this seminar series. Let me try to share my slides. All right. Okay. Does that work?
It's coming up.
Oh, why is that my last slide?
Yeah, they're coming up.
But,
Yes, it's
yeah.
working.
All right. Do you see my first slide?
Yes, first try, yes.
Okay, all good. All right, so I will very briefly tell you, I only have 10 minutes, about some of the work we've been doing in my group at the Pasteur Institute and also in collaboration with Eligo Bioscience, which is a startup company based in Paris that I co-founded and that is focusing on the development of CRISPR therapies targeting the microbiome. So as a quick introduction to CRISPR tools, there has been tremendous development of different CRISPR technologies over the years, giving the ability to do all kinds of genetic perturbations in bacteria. So it of course started on the left with CRISPR nucleases that you can use to cut the chromosome of bacteria. And we quickly realized that when you do that, the main outcome is actually that you kill the target bacteria. But you can also deactivate, for instance, the Cas9 protein so that it binds to the target sequence but doesn't cut. And in that case, you can bind the target gene and block its transcription. This is what is commonly known as CRISPR-I. You can also recruit transcriptional activator domain to promote the expression of target genes. Importantly, a very nice editing strategy which was invented by the group of David Liu, is this base editing method where you fuse a deaminase to a dead Cas9 or a Cas-nickase. And in that case, what you do is just a chemical modification of a single base at the target locus, which is a very nice way and efficient way to do genetic modification without having to introduce double-strand breaks. Beyond that, there are even newer CRISPR tools that people have been developing and keep exploring. Very interesting set of tools are what we call CAST for CRISPR-associated transposase. So those are derived from natural transposons that use CRISPR-Cas complex to guide transposition events, and they can very nicely be reprogrammed with guide RNAs to direct insertions of large DNA payloads at any target site of choice. And you also have more other fancy editing methods, including prime editing that I won't mention too much here. All right, so my lab has worked a lot over the last 10 years on the characterization and development of mostly the CRISPR-I techniques, so where you use a dead Cas9 to silence genes. And one of the things we've been doing, I want to give you a brief snapshot, is developing high throughput screens with CRISPR-I, where you can design large guide RNA libraries targeting every gene in your chromosome. You order them from an on-chip oligosynthesis, you clone them into your plasmid of choice, and then you introduce them into your bacteria to start the screen. And so the way those screens go is that you induce the expression of your dead Cas9, and you sequence the composition of your library both at the beginning of the experiment and at the end of the experiment. And so what happens is that guide RNA that silence some genes that are essential for the growth of the bacterium will be depleted and lost during that screening, while if a guide RNA provides a fitness advantage, it will be enriched during the screening. You can then infer in just the whole pool the fitness impact of every gene in the population. So we've done over the years a number of those screens, and they've proven extremely powerful to decipher bacterial genetics in high throughput, and it's a technique that is now quite broadly adopted. And I just want to show you some very recent work that we're doing now to apply these technologies in the context of the gut microbiome. And this is work that was done by an amazing PhD student, Amandine Maire, in collaboration with Nathalie Royon in Paris. And so Amandine has constructed guide RNA libraries targeting different E. coli strains and introduced them by gavage into the mouse gut. And the same story, we monitor the composition of the library, and this can let us look at the impact of different genes on the fitness, that is then the ability of E. coli to colonize this gut environment. And we've been using this to study E. coli colonization in the gut in different conditions, including looking at different diets. I said looking at different strains of E. coli, and also looking at healthy versus inflamed gut environments. So I, of course, don't have time to tell you everything we've been doing here. I'm just giving you just a very quick snapshot of one experimental result here. So this is an experiment in which we give different groups of mouse different diets. And here it's a genome browser view, so that you get a sense of what the raw data looks like for such a screen. And here we're looking at this plot where every point on this scatter plot is just the effect of a different guide RNA in a different animal. And you see in green are the animals that receive the high fiber diet. And so the other animals, there is basically no effect of silencing this fructose utilization gene. But you see that when you block the expression of fructose utilization in the high fiber diet, it's very detrimental for gut colonization. And actually what turns out is that what happens here is that in the high fiber diet, there is a lot of inulin. And there are bacteroides in the mouse gut that can bloom in this inulin diet. And when they utilize inulin, they release fructose in the medium, and then E. coli that can use this fructose do better than if they cannot use this fructose. So this is just a very quick example showing how through this kind of high throughput experiment, we can learn a lot of things about the genetics, but also about the environment, about cross-feeding interactions that can happen in the microbiome, and more. Another very quick snapshot. This is results from a screen we did in healthy versus DSS-induced inflammation in the animal. And so this is just a different way of representing the data with a PCA plot on the left here. And what you see here is that each point on this plot is just a different animal. And in red, what you see are the animal that received the DSS, so that were with the inflamed gut, versus in blue, the healthy animal. And you see that there is a very nice separation of that, meaning that actually the results of the CRISPR screens, you could almost think of them as a kind of diagnostic tool that give you a very rich picture of the environment in the gut. All right, for the sake of time, I'll move a bit to another very quick story, which is to show you how now we're thinking about not just constructing this type of genetic perturbation in vitro in the lab and then reintroducing it in the mouse gut, but we think it would be extremely powerful if we were able to directly do the genetic perturbation in the bacteria directly in the gut environment. Of course, to do that, we need a good way to deliver CRISPR-Cas system to the bacteria that are in the gut. And for this, we're actually turning to phage meats. That's to say, plasmids packaged into phage capsids that we can then deliver to the gut environment. And so for this, we've been relying a lot on tools and things developed with Oligo Bioscience. Was focused a lot on the development of these phage meat vectors to deliver different things for therapeutic application, which includes the delivery of CRISPR nucleases to kill specific strains that might carry virulence factors, toxins, antibiotic resistance genes. You can also deliver base editor to do just targeted point mutation and inactivation without killing the bacteria, or you could think of delivering even therapeutic genes to be expressed directly by the bacteria in situ in the gut environment. So I just want to give you a brief snapshot of a recent study we published with Oligo Bioscience, in which we demonstrated the use of base editor for in situ genetic modification. And so to deliver this, we needed to engineer an efficient delivery vector, and we turned to phage lambda, and we had to engineer the tail of phage lambda, so the tail fiber, the tail tip protein, so that they would recognize receptors that are actually expressed and available in E. coli when it colonizes the gut environment. And through this engineering, we were able to obtain efficient delivery, and we've been delivering different kinds of base editors, either adenine base editors, cytosine base editors, to modify or inactivate genes directly in the gut. And so here is just an experiment to show you that this is possible and actually works. So we colonized here the animal with just the MG1655, that's a model E. coli strain we use in the lab, but in which we had an ampicillin resistance gene. And we could monitor the inactivation of that gene after delivering either a single dose of this phage meat delivering the base editor, or we can also try multiple doses over a few days and see what happens. And what you see is that if you give a single high dose, you can already edit around 90% of the target bacteria directly in the mouse gut. And if you give multiple doses, this is a slightly lower dose, so you see increased editing rates as you keep giving more doses. But quite interestingly, after we give the last dose, we kept track of the animals over 6 weeks, and you see that the edit remained very stable over the whole duration of the experiment. So this is a very interesting proof of concept that it's actually possible to genetically modify a target bacteria directly in the mouse gut, which I think opens a lot of research directions for both for research and possibly also for therapeutic interventions. So last word, because my time I think is up, is just to give you a snapshot about, I think, exciting research directions that my lab is taking at the Pasteur Institute, where we study to work quite a bit on fascinating genetic systems called diversity generating retroelements. And so these genetic elements are actually carried by some bacteriophages, and they were first described in the early 2000s by someone called Jeff Miller at UCLA. And what these DGR systems do is that they introduce programmed high permutation in variable loops of the tail fiber of the phage. And so this enables the phage to accelerate its own evolution so that it can very quickly bind new receptors and infect new host bacteria. And the way this works is that you have this variable region and you have a template region, this TR sequence, that's part of a non-coding small RNA. And this non-coding RNA is reverse transcribed by an error-prone reverse transcriptases that make mistakes, in particular at adenine position, producing a mutagenic cDNA that can then retrohome and replace this variable region. And so it's been said that among the known biological mechanisms for genetic diversification, DGRs are capable of exploring the highest ceiling on coding sequence variability. So I think those are absolutely fascinating systems. And we've now recently been able to harness them in E. coli. We can redirect them to any sequence we want. And I'm very excited by what we will be able to do with this. And so with this, I'd like to thank you for your attention. A quick thank you to the people in my lab at the Pasteur Institute, to the people at Eligo Bioscience. And with this, I will leave the floor to our young investigators.
Thank you so much. This is amazing. You know what? You always kind of starting something new. I mean, including CRISPR and also, applying CRISPR technology into the microbiome. And both field is only a little bit more than a decade old. So this is a fascinating. And last part is also very exciting because that potentially open up new field or new technology, new tool for a lot of people, including metabolic engineer, also microbiome, researchers, I guess. So thank you so much for your pioneering, really pioneering work, and then we were happy to use those kind of tool in the future again. Thank you so much.
Yeah, thank you.
Yeah. All right. Now the main speaker of today with a longer introduction. Professor Yaojun Tong is a tenure-track associate professor at Shanghai Jiao Tong University. He just started, I believe, so congratulations, starting new position. He is a recipient of the NSFC Excellent Young Scientist Fund and Shanghai Overseas High-Level Talent Program awardee. He has long been engaged in microbial synthetic biology research, specifically focusing on key microbial chassis such as E. coli, Streptomyces, and yeast. He has systematically established a suite of highly efficient genome editing platforms, including CRISPR-Cas9, CRISPR-I, CRISPR-Best, CRISPR-N-Race, and also the ultra-compact TnpB-based stage system. So this is today's common, topic, CRISPR technology. He pioneered single-base and multiplex-based editing in Streptomyces. Streptomyces is not easy bacteria to deal with because I know 30 years ago I actually working with the Streptomyces. I don't want to touch that bacteria again. This is too difficult. But he developed a lot of tools in that, Streptomyces. and also advanced the first systematic application of lightweight TnpB editors. His work has been published in big journals like PNAS, Nature Communications, Nature Protocols, and other leading journals. He has led multiple major projects, including the National Key R&D programs and also NSF grant, filed more than 10 patents, and made genome editing tools freely available to over 200 institutions worldwide. He also serves as associate editor of Synthetic and Systems Biotechnology and Frontiers in Bioengineering and Biotechnology, a committee member of International Metabolic Engineering Society, in short IMES, Early Career Committee, chair of the Metabolic Engineering and Biomanufacturing Committee of the Chinese Society for Microbiology, and secretary of the Molecular Microbiology and Bioengineering Committee. He also served as an international reviewer for Netherlands Organization for Scientific Research, in short NWO. I especially thank him for joining us at this inconvenient time. At that, right now China is past midnight, and also just before the biggest holiday. And also I heard he is on vacation right now, starting today, because this is past the midnight. So Yaojun, thank you so much again for your time today, especially after midnight. And please take it away, all yours now.
Okay. Can you see my screen?
Yes, I see your screen.
Okay, cool. And thank you so much. If I pronounce it not said correctly, please apologize. So Taisu, is that right?
Yeah, my name is Tessa, but people call me Moon.
Okay. Yeah, so for the invitation and also the nice introduction. So hello everyone, and good morning, as you said, good evening, and also good afternoon, depending on different time zones that you are. So in China, just like you mentioned, it's 13th of February now. So I'm very happy today to be here for this wonderful SynBYSS talk. So I'm Yaojun Tong from Shanghai Jiao Tong University, as you just mentioned, and today I'm going to give you a quick through of some recent works that I just doing ongoing and finished in the lab in recent years. And my talk today will focus on two different parts. One is the genome editing toolkit development, and then the gene regulation systems that we are working on for bacteria, of course. And let me start with a quick overview of my lab, named as SynBYSS Lab, that we are doing actually three things. So we work on both of the fundamentals of and also the applications of synthetic biology. On the application side, we engineer smart microbial cell factories for different things. For example, living therapeutics, and for production of metabolites. On the fundamental side, we study microbial social behaviors, so-called social microbiology, and we are decoding that how bacteria they can communicate with each other, especially for a kind of very difficult to manipulate bacteria stomatitis. And to bridge these two, we built enabling tools, especially genome editing and gene regulation tools. So today I'm going to only focus on the enabling technologies we are developing in the recent years. So before I begin, I want to thank all my students, our collaborators, and our funding agencies. None of this would have happened without them, and I'm really grateful for their supports. Of course, I also want to thank this very beautiful bacteria, SynBYSS. They make our science possible and keep it fun. They have given us many of the antibiotics that we are currently using in clinic, and also many very important anti-cancer drugs. Of course, they also deliver a lot of bioactive compounds for many other purposes. However, after the golden age of 1950s, 1960s, the pipeline of natural product-based drug discovery, antibiotic discovery is drying up. But based on the genome mining, scientists found that more than 90% of the gene clusters that encode secondary metabolites that we are using for drugs have not been assigned with any known compounds. So that means we have a lot of potentials to find new compounds out of this bacteria. To make this potential a reality, we need, higher ladders for these high-hanging fruits, because those easily to be found ones already be discovered. And before CRISPR, the only way that can manipulate the genes of SynBYSS is PCR targeting, and that is a very complicated experiment setup, just like this slide shows. It's very complicated. I'm not spending time on explain the details. Then, thanks to the pioneers, just like David today with us, CRISPR pioneers, they bring us the CRISPR-Cas9, the very wonderful tools that we are using for manipulate genes, genomes of almost all kinds of organisms today. And we are now entering a new world of engineering of this bacteria. And to me, I want to see that genome editing, actually there's, let's say, a CRISPR-Cas9, there's no real magic. So the only magic that I can see is how to repair the double-strand break, because all those genome editing events actually happened after how how the cells decide to repair the double-strand break, or the nicks that caused by the decays. And normally there are two ways. One is the non-homologous engineering, and the other way is HDR. But very interesting, very interestingly, in SynBYSS, there's a third way. It's a defective non-homologous engineering. That means that it has partially of the activity, but not fully functional non-homologous engineering, and it can make a random size deletion in between two essential genes. So you can think of that if there's double-strand break, and the cells want to clean up all those damaged DNA, and those exonucleases want to digest the damaged DNA. And in the meantime, the bacteria, the cells want to survive, so the ligase want to make the break repaired. And so, if this break be repaired before touching the essential gene, so it can survive. That means that we can have, if we picked enough clones, so we can have a library of random size deleted clones, and that can do some very interesting survey and study of the essential genes and metabolites related to different pathways. And to sum up of this CRISPR-Cas-based toolkit, we started the end of 2013, and that is already 10 plus years. We developed a lot of different tools for SynBYSS, also later for E. coli and also yeast. We now can do double-strand break DSP-based genome editing. Also, we have CRISPR-based, so the single base editing systems, and later we have this prime editing as well. And we freely distribute our toolkit, our tools to more than 300 different labs worldwide. And also we got this Blue Flame Award. I think that is name from Yaojun, and I think that's the one contribution that we make to this society. So from PCR targeting to CRISPR-Cas mediated genome editing, we achieved a lot of very good, important advantages. For example, CRISPR-Cas9 can make genome editing much faster, and also with high efficiency, easy handling, and also precise. To date, I think CRISPR-Cas9, at least I believe that CRISPR-Cas9 is still the best choice for genome editing, but still there are rooms for improvement. For example, the size for CRISPR-Cas9, Cas12 is relatively large, and also because the activity is very strong, and in the meantime also can cause some side effects, so-called cytotoxicity. And also the native Cas9, the target recognition pattern is GC-rich palm, and that's for some reason is also can be one of these limitations that for different position targeting. And then we are focusing on another very compact and small in the nucleus, TnpB, and that is the ancestor of Cas12. The size is only 408 amino acids, 1/3 as large as CRISPR-Cas9. And TnpB so far is the most compact in the nucleus that have been reported with genome editing activity. We investigated 3 different compositions of TnpB omega RNA systems. Only the one separately expressed TnpB and the omega RNA showed gene editing potential in vivo. So based on this composition, we built a stage, the SynBYSS compatible TnpB-assisted genome editing toolkit together with Professor Ampli from KAST. And we use the model organism, SynBYSS Silicola, as the test strain. And we're targeting 2 different key enzymes from this actinirodin biosynthetic cluster. If one of these 2 key genes is inactivated, so the phenotype of this strain will show non-blue, because this actinirodin compound is blue and it can diffuse to the media. That means if you have this cluster in and alive, So after like 5 to 7 days incubation, you can see that the plate will be dark blue. But if this compound is no longer be produced, so the phenotype will show a reddish color. And we use this phenotype change as our indicator for quick screen of our system. In the meantime, we established 3 different subsystems for different DNA double-strand break repair pathways. Here is how we do the evaluation of these 3 different subsystems. First of all, we randomly pick those non-blue clones. Then we subject them to PCR and then SNR sequencing. And we calculate the different editing efficiencies based on different DNA double-strand break repair pathways. The editing efficiency across 30% to 70%, and that is much lower than CRISPR-Cas9. For natural products discovery, we routinely need to deal with biosynthetic gene clusters, which can be 100 kilobases. Our stage system also has the ability to delete BGC size DNA fragment. For example, this actinorodine BGC is 17 kb-ish, and this red BGC is around 30 kb, and the editing efficiency is around 20 to 40 something efficiency, and that is acceptable, not that good, but acceptable. Also, next we use the whole genome sequencing based method to evaluate the off-target effect. And very interestingly, the cases that we tested, we didn't find any TnpB-associated off-target event. And that means it is very fidelity, and the specificity is better than CRISPR-Cas9 based tools for actinomyces, for streptomyces. And next, as you know that streptomyces has a very unique feature. So it has a linear chromosome, not like E. coli, it's a circular chromosome. So linear chromosome means if we want to deal with 2 genes, or sometimes it's just another copy of 1 gene, so it's identical sequence. And then if we make a double-strand break, that means there will be 2 cuts, and that cause very severe cytotoxicity to this organism. And for some cases, we just cannot get a very clean knockouts. So we want to move from DSP to DSP-free system, and that comes CRISPR-based, so this best editing system. And in our previous CRISPR-Cas toolkit establishment, we built this CRISPR-based, so which includes 2 base editors. One is CBE and the other one is ABE, so the very original one. So in particular, C2T editing can generate nonsense mutations by converting 3 codons, tryptophan, arginine, and glutamine codon into stop codons, leading to gene inactivation. And by optimal experiment setup, we now can inactivate a gene within 7 days. So it's very quick. So similarly, we also implanted base editing system to our stage. And because the vehicle TnpB nickase somehow is different compared to Cas9 nickase, and it makes the base editing window slightly different. So for TnpB, the window is 2 to 12, and with the peak activity at position 3 to 7, and the overall efficiency is compatible to Cas9 nickase mediated base editing system. And as metabolic engineers, we often need to deal with multiple genes at a time. So we move from single gene targeting to multiple gene targeting. One particularly attractive feature of TnpB system is that its guide RNA can be franketed by hammerhead ribosome, and we self-cleaves to generate processed guides. And this built-in RNA self-processing makes TnpB naturally well suited for multi-gene targeting. Consistent with this, when we programmed the system to target 2 genes simultaneously, we achieve robust editing with good efficiency, as you can see the right lower corner. So as I mentioned earlier, TnpB generally edits less efficiently than Cas9, so which also means there's plenty of rooms for improvement. In collaboration with Professor Hong Liang, we applied a protein engineering workflow guided by a large language model. And we successfully obtained TnpB variants that can achieve nearly 100 editing efficiency. So the take-home message here is, at least, so for me, I'm not a computational biologist, so I actually honestly don't know the details what happened inside, but the reality is we got the desired mutants. So take-home message here is that, at least in our case, this AI-guided approach can outperform experience-driven human rational design approach. So like these 2 circuit mutants, that if ask me to rational design, I will never touch these two domains, because they are far away from this active pocket RUVC domain. So that is for the nucleus activity. But the computer gives us surprising results. That is not from this RUVC nucleus domain. So this suggests that the model is capturing long-range effects that are easily to be missed by experience-guided approach. So I think AI is great for protein engineering studies. So next, to facilitate omega RNA design, we collaborated with Kaibling and Timman at DTU, Technical University of Denmark. We upgraded CRISPR Web to version 3, and now it supports TnpB omega RNA design. Please feel free to try it. I think this is very good Web 2 that can make, first is that it has visualized interface, and second is it allows users to upload their own genomes. So the design pipeline works directly in their student background. And a quick sum up of the stage toolkit. So now we have multiple subsystems that can deal with both DSB and DSB-free genome editing events. And now let's switch gears to gene regulation. So as I like to say, normally regulation brings order. It's just like traffic control. If there's no rules, no signals, that means it will be very chaotic and less efficient. Just like the same concept for metabolism. Gene regulation is what organizes, flex, and coordinates multiple pathways together towards our desired outcomes. So the central dogma tells us that going from gene to protein, it has two key steps. One is transcription and then translation. So today I'm going to focus on translation regulation. In bacteria, translation has several distinctive features. One is that transcription and translation are tightly coupled. Then the second is that they normally organized as an operon, means that several genes, they chain together and controlled by only one promoter. And there are few tools are available for directly regulating bacterial translation. So we want to make this regulation more efficiently, and also can deal with single gene orthogonally within an operon. We aim to harness CRISPR-Cas13d as a programmable platform for this purpose. So CRISPR-Cas13 is an RNA-guided RNA targeting system. Upon activation of its RNA activity, Cas13 can trigger its collateral activity that can cleave bystander RNAs, which often cause toxicity to bacteria. We found that wild-type Cas13d and some reported high-fidelity variants showed strong toxicity in E. coli. But the high-fidelity variants indeed displayed increased specificity, but still the toxicity is a big issue. We propose an engineering hypothesis that balances Cas13 activity, targeting specificity, and serotoxicity. Then we can obtain this desired mutant that we want through a rational protein design. So what we were doing is we want to first is to reduce the toxicity and to maintain the specificity and the activity. So what we want to do is try to shrink the active pocket, and by doing that, indeed we got some variants that can meet our design. We name them attenuate Cas13d. So in short, ATN Cas13d. So as you know, Cas13d also can bind to the target, but it lost its cleavage activity. Just sit there to block the ribosome loading. So it mainly causes translation inhibition. So the downside is that this creates two separate systems. So the active Cas13d for knockdown and the Cas13d for translation control, which increase the complexity. because now we need to use different systems to do different tasks. So interestingly, our attenuated Cas13D mutant provides a middle ground. It keeps enough RNA activity for strong transcript degradation, but with reduced collateral damage and thus much lower toxicity in bacteria. Functionally, Cas13D gives a precise translation block with a narrow targeting window, mainly around the RBS and the start codon position, which is ideally for gene-specific control in operons. In construct, attenuated Cas13D degrades the transcript and shows a broad, effective targeting window, which robustly knocking down across many sites along the mRNA. So this is a very interesting mutants variants. On this slide, we show that we can decouple binding from cleavage by just a simple engineer of the CRISPR RNA. With the perfectly matched CRISPR RNA, attenuated Cas13D is activated, and we can see mainly transcript degradation. So both reporters in the operon drops together. But when we introduce 5 proximal mismatches in the CRISPR spacer, we can progressively weaken RNA's activation while keeping RNA binding. In the saying, attenuated Cas13D behaves more like a translation blocker. It sits on the target mRNA and inhibits translation without broadly degrading the transcript. So we screened a mismatch series from PM1 to PM12, as you see from here. Clearly, there's a pattern that from PM5 to PM7, the target SFGFP is strongly repressed, while mCherry is largely maintained, indicating that RNA's activity is largely abolished, but binding and translation inhibition are preserved. So by tuning a few proximal mismatches, we can switch Cas13D from an RNA degradation mode to a more precise translation regulation mode. So to now, our system can make targeted targeted translation reparation. And next, we want to add the function of translation activation into the same system. As our lab believes that less is more when it comes to creating new methods and tools, just like an old Chinese saying, 大道至简. So we need to make the tools as simple as possible, and then it can make users much easier to manipulate and to access the tools and technology. What we did is to fuse the translation initiation factor IF3 to our attenuated Cas13D, and combined with this PM7 CRISPR RNA structure, the system can activate the translation of targeted gene within an operon and not influence the other genes in the operon. And this slide shows that attenuated Cas13D IF3 complex acts as a multifunctional RNA regulator in bacteria. By targeting the 5' UTR, you can see from the left corner, the IF3 fusion actually does not distribute the original Cas13 behaviors. With PM7 CRISPR RNA, we can still get translation inhibition. and with the perfectly matched CRISPR RNA, so the attenuate Cas13D can, let me see, we're here. So yes, so if we use a perfectly matched CRISPR RNA, we can get this transcript degraded. And then we can program the inhibition, activation, and transcript degradation in parallel using different CRISPR RNAs. Only by changing the CRISPR RNA, we can achieve different genome editing events. The main constraint is target abandonment. Activation and the inhibition, sometimes they have this crosstalk and they have this cross interference. But if we are dealing with medium or low copy number genes, so the three regulatory models become much more orthogonal and clean. We give this system a very cool name, SONAR, so synthetic orthogonal nucleic acid activation and reparation system. We also use this. We try to see if this SONAR system can work in industrial scale fermentation. So we use the lycopene production to demonstrate the SONAR system actually works in this industrial 3-liter fermenter. We found that some genes, they are essential, but they can heavily influence the lycopene production, and those genes can never be identified or screened out by a traditional method. Now I would like to quickly summarize the unpublished SONAR system. Long story short is that we construct one platform, and this platform has 3 modes. Can simultaneously, by changing CRISPR RNA to achieve translation activation, translation block, and transcript degradation within one operon. And here comes the end of my talk today, and I want to quickly sum up. So first, we developed CRISPR-Cas9 based and non-CRISPR-Cas based TnpB genome editing system for osteomyces. And then we also developed the SONAR system. Hope that this story will be published soon. And if you're interested, you can read the paper later. Before QA session, I also have 2 quick advertisements. Just as Moon says that I'm associate editor of Synthetic and Systems Biotechnology Journal, and if you have related works, please submit. And then the other announcement is that we have this International Synthetic Biology Conference in the end of March 2016, and please scan and visit our website and to submit your abstract for participating in the conference. And that's all. Thank you for your attention, and now I'm happy to take questions.
Thank you. This is amazing work. The amazing, you I feel like you are not anymore rising star. You already risen. This is amazing. So, David, I now see your, the time is, evening, because I'm seeing the dark outside. So, do you have any comment or question?
It's very cool. Very nice to see how you've been expanding the toolbox so much now. Very nice Cas13 tools. It's the first time I hear about activation of translation, which sounds like it could be useful in many cases. How do you see actually translation activation versus transcription activations? What are the pros and cons for you?
Yeah, that is a very good question. So thank you, David. And for because we are targeting mRNA, right? So it's Cas13 system. By literature reading and by the knowledge that we have, I don't think the Cas13 CRISPR-RNA complex can have effect on DNA level distribution.
No, for sure.
So,
I had more in mind some other tools that people have been developing for transcription activation, but I think transcription activation in bacteria where people fuse dCas9 with different activator domains has
that's
definitely a lot of
difficult. Yeah. So you are the pioneer. So 2013, this NAP paper. So this
right.
dCas9 fused to omega subunit, and then there are several other papers to see that
Yeah.
dCas9 to fuse to different this activation domain. But honestly, in bacteria, in the lab we tried. It depends. So case dependent. So in some cases, in some very low activate low active promoter driven genes, the effect will be better than those high expressed genes, and also different activation domain for this transcription. They somehow the behavior are different. Long story short,
Yeah.
it's not that stable. So
Yeah.
from my observation, and for translation manipulation or translation activation, what we were thinking is we want to jump to another layer of this regulation or control, because just as I said, from DNA to mRNA, so this transcription step, somehow the current available tools are not doing that well. So we want to jump out to just manipulate mRNA to protein step for the translation.
And so does that do you see an activation of translation only when you have a very bad RBS in front of your genes, or do you also see it if you already put a good RBS?
We didn't do that yet. So now we only focus on the RNA to protein step. So we didn't investigate and didn't see the transcription side.
So yeah, I wasn't asking about transcription. I was asking about the ribosome binding site. So I was wondering how the activation by your Cas13 IF3, how is it impacted by the strands of the ribosome binding site for that transcript?
That's a good question. Yeah, let me just quickly have a look. Yeah, so we need to make the system in a range of distance relatively to the position of IBS. We need to let me just see where it is. Okay, here. Yeah, so different positions, they have different effect, and too close to RBS can somehow reduce the activation activity.
Yeah, I wasn't really asking about the distance, more about whether you tested different RBS and see if that changes the activity.
We
selection.
are doing that.
Okay.
So far, we didn't find a very good correlation among different IBSs. Actually, we are testing, and also we are testing different abundance of this mRNA by using different promoters to make the system with different amount of mRNA and to see how the activation actually varies. Yeah. Hope that we can have some results later on. We are testing. Yeah, thank you.
Okay, Dave, do you have any other comment or question?
No, all good for me. Again, congratulations on all the nice work and tool development, and hope we get a chance to meet in person soon.
So now I see the Dave need to go for dinner soon. Let me ask one quick question, but very challenging question to both of you, because you are talking about tool development. And when I was the master student almost 30 years ago, I working on streptomyces, then especially the S. silicola and some other streptomyces, and then at that time we didn't have the mini prep kit, so I need to do mini prep by hand and manual extraction everything for 3 hours just to do mini prep. And but now we are talking about CRISPR revolutionize the scientific community, and then we now talking about AI potentially revolutionize the entire protein engineering and some other thing. Let me ask you one quick question. So you have been developing a lot of important tools, but let's say from today and the 10 years later, if you wanted to have one tool developed, what would be that one tool that potentially revolutionize entire field, biology, biotechnology? Just one. I mean, other than AI, other than CRISPR, other than what would be that tool? I mean, I'm just curious.
Yeah, that's a great question. I will need some time to think deeply about it. What is the biggest kind of pain point that we currently have in experiments, or what are the main bottlenecks? I mean, of course, I mean, you said no AI, but of course, if we had the ability to design a new protein and that work as expected from scratch, or new genetic circuits that work from scratch, yeah, that'd be amazing. And I think AI will play an instrumental role there for sure. I think then if we talk more about manipulation tools, genetic tools, I think the toolkit is really getting better and better. There are still some time-consuming steps in some of those things we have to do. So I think if we could just make things even more convenient to get DNA in bacteria. I think, yeah, one thing is actually that all these tools tend to work very nicely in model bacteria that we can manipulate easily. But when you try to bring them to non-model bacteria, gut bacteria, etc., then you're in for a lot of trouble.
Yeah.
And so I think that's definitely one of the next step for this field in general, and people are starting to do this, but it's bringing all this magic now to other bacteria that are important for our health.
Sure, sure. How about Yaojun? Do you have any one tool that developed in that way your research is so easy? 10 years.
Yes, so I cannot agree more with David. So like we are now doing manipulating different bacteria, not even mention different organisms. Even like you just mentioned the stomatids, like different stomatids, we can not just use one system to do genome editing. We need to do a lot of optimization, a lot of different parameters are changing. So for me, I think if I can only have one opportunity to have one tool, I think I want this unified general tool that can manipulate DNA.
Yeah. So
Not
I feel the similar
not
way.
different.
Yeah.
Yeah.
Yeah, I feel the similar way, but one thing I want to see is 10 years later, if I wanted to just kill one bacterium in the microbiome, and then regardless of the whether there's a non-model organism or whatever, I can kill. That means that's a knockout, one single strain knockout. If you are going to add one bacterium without any problem into the community or the microbiota, we can do that. So I want to see also even so Dave, you started already because you showing the in Nature paper, you can do in situ manipulation of whatever bacteria. But I want to kind of see any bacterium you wanted to manipulate, and then we can do.
Yes, that's the dream. I agree.
Yeah,
That's
that's
the
the dream.
dream. I think
And for you,
we
you
should.
started already. You started. Jennifer Doudna already started. I also starting to do so, but still we are very limited. We only doing E. coli,
Yeah.
Pseudomonas, or of a handful of the bacteria right now.
Yeah, still lots of hurdles to overcome, even things we don't know we need to overcome that we'll have to overcome. So
Yeah.
that's what's exciting about it is also that when we try to do these things, we also learn new biology.
Yeah, so I believe because of that, I was so excited when you started the conference about, genome editing, microbiome editing conference, because that is what I wanted to see. 10 years later, we're talking about in the conference, we're talking about we able to manipulate hundreds of the bacteria without any problem. I want to see that 10 years from
Yeah,
now. Okay.
yeah, that's a good goal.
Yeah, I realize I need to let Dave go
Yeah,
for dinner, and also Yaojun need to sleep because it's
yeah.
the past one
It's
20.
already quite amazing. We managed to have a speaker in California, one in France, and one in China doing a call together. So yeah, congrats on putting
So
this
cool.
together.
So
Yeah,
cool.
so thank you so much. I will close, by saying something. So thank you all for joining and staying today. We'll meet again on February 19th, Thursday, the same time, the same Zoom link. We'll have the Professor Jean Michel on at, I think, originally from also France at the University of Wisconsin-Madison, and Professor Rosa Santomantino, I think originally from Italy, at Cornell University. As usual, the following formal chat will occur without recording. Please stay here if you are interested in chatting with us. Thank you, and I stop recording. 1 second.